In the last video, I described DNA sequencing by the chain termination or dideoxy method, also called Sanger sequencing. That method works well for some applications, but has limitations. A sequencing method developed in 2006 and currently marketed by the company Illumina gets around some of these limitations. First, for Sanger sequencing, you need a fair bit of template DNA, so the DNA must be amplified in some way. You can do this by cloning into a plasmid and having bacteria make a lot of the DNA, or by PCR. In Illumina sequencing, less DNA amplification is necessary, and the amplification step is built into the technology. The basic idea is that a mixture of DNA fragments is deposited onto a solid surface, such that individual fragments are located on random places on the surface. Then, copies of these fragments are made in a process that resembles PCR. You end up with clusters of DNA strands, with all strands within a cluster identical to each other, but each cluster different from the others. The surface is large enough to accommodate millions or even billions of individual clusters of DNA strands. You can therefore analyze a very large number of DNA strands at the same time. Illumina sequencing is said to be massively parallel for this reason. In contrast, in Sanger sequencing, each template needs its own sequencing reaction and capillary electrophoresis, making it hard to scale up the process beyond a few hundred simultaneous reactions. The electrophoresis step of Sanger sequencing is also time-consuming. In Illumina sequencing, no separation of strands is necessary, and bases are read in real time. The result of all these improvements is that sequencing large amounts of DNA is faster and much cheaper than it would be using Sanger sequencing. The first human genome was sequenced using Sanger sequencing at an estimated cost of about $450 million to produce the finished sequence. Using modern technologies, the cost of sequencing a human genome today is considered to be $1,000 or less. One drawback of the Illumina technique is that the read length, or the number of consecutive base pairs that can be sequenced from a given template, is about 150 to 300 base pairs, which is shorter than the read length for Sanger sequencing. Now I want to go over how DNA sequence is actually obtained in the Illumina method. Remember that, after the amplification stage, we have many clusters of DNA sequences attached to a solid surface. This sequence here represents one of these clusters. During the preparation step, a common end sequence is added to all of the strands. To sequence the DNA, a primer is added that will anneal to this added end sequence on each strand. You then incubate the surface with DNA polymerase and a mixture of all 4 dNTPs. Each dNTP is labeled with a different fluorescent group, as in Sanger sequencing. Each dNTP also has a removable blocking group that blocks the 3' hydroxyl of the nucleotide so that only one base can be added to the primer. At each strand, DNA polymerase will add the appropriate base to the end of the primer using the existing strand as a template. In this example, there's C on the template, so a G is added. Because of the blocking group, only one base is added. Then you wash away the unused bases and measure the fluorescence signal of the group that remains. In this case, a yellow signal would be seen. Then you remove the fluorescent tag and the blocking group from the newly incorporated base. You wash them away and you have extended the primer by one base. Then you add DNA polymerase and the fluorescent bases once again, and another base will be incorporated into the growing strand. In this example, it will be C. Excess bases are washed away, the fluorescent signal is read, and then the fluorescent group and blocking group are removed, and the cycle starts again. This figure shows what you would see if you were looking down on the surface. Each one of these black boxes represents a different sequencing cycle. Looking down on the surface, you will see a bunch of fluorescent dots. Each dot represents a different cluster of DNA strands. The color of the fluorescence indicates the base that was added in that cycle. So on this top circled dot here, it is green. That represents the base C. In the next cycle, This position has a blue dot, meaning that A was added. In the third cycle, the dot is red, meaning that T was added in that cycle. The computer simultaneously collects data from millions of spots each cycle, which tells you the sequence of millions of DNA strands at the same time. Remember that the reads are only a few hundred bases in length, so if you want to get the sequence of a whole genome, you need to assemble the sequences properly. I'll talk about that in a future video. But for now, having explained how we can sequence DNA, I want to get back to the final stage of cloning your favorite gene into E. coli, overexpressing and purifying the protein product of the gene.